The ocean floor, 550 million years ago, was covered in slime. Thick microbial mats, living carpets of bacteria and algae, blanketed the seabed. Large, soft organisms sat motionless on this surface, absorbing nutrients through their skin. No mouths, no guts, no eyes, no predators. Life was quiet, still, and profoundly alien. Then, in a window geologists measure at roughly 20 million years, everything changed. Animals with shells appeared, animals with eyes, animals with claws, guts, and nervous systems capable of coordinated hunting. The oceans transformed from a passive, absorptive world into an arena of predators and prey. Every major animal body plan alive today, arthropods, mollusks, vertebrates, was invented in this brief span. This was the Cambrian Explosion, and it didn't just add complexity to life, it redefined what life could be. For 3 billion years, Earth belonged to microbes. Bacteria and single-celled organisms dominated every environment. Multicellular life appeared late, sometime around 600 million years ago, but it bore almost no resemblance to the animals we know. The Ediacaran period, from roughly 635 to 541 million years ago, was the world immediately before the Cambrian. It began after one of the planet's most extreme ice ages, an event geologists call Snowball Earth, when glaciers may have extended all the way to the equator. When the ice finally melted, it released a massive pulse of minerals into the oceans, phosphates, iron, and other nutrients that had been locked in frozen rock. The seabed during the Ediacaran wasn't sand or mud, it was a microbial mat, a living surface built by layered colonies of photosynthetic bacteria. These mats were sticky, cohesive, and covered vast stretches of shallow ocean floor. The organisms that lived on them were equally strange. Dickinsonia was a flat, ribbed, oval-shaped creature that could grow over a meter across. It had no head, no tail, no obvious organs. For decades, scientists debated whether it was even an animal. In 2018, researchers found cholesterol molecules preserved in its fossils, a chemical signature unique to animals. Dickinsonia likely crawled slowly across the mat, absorbing food as it moved, leaving faint trails behind. Charnia looked like a large fern or feather, anchored to the seafloor in deep, dark water far below the reach of sunlight. It couldn't have been a plant. The best interpretation is that it was an early animal, filter feeding from the current, passively collecting microscopic particles. Kimberella was different. It had a slug-like body and what appears to have been a feeding tube or proboscis. Fossils show scratch marks around it, suggesting it actively grazed on the microbial mat. This implies a primitive gut, a more animal-like metabolism, and behavior more complex than simple absorption. But these were outliers. Most Ediacaran organisms had a quilted pneumatic construction, segmented bodies that looked inflated, like air mattresses stitched together. They had no mouths, no anuses, no clear sensory organs. They sat still. They absorbed nutrients. They left almost no evidence of predation, competition, or conflict. Oxygen levels during the Ediacaran were low, perhaps 5 to 10% of modern atmospheric concentrations. The deep ocean was anoxic, lacking oxygen entirely, and rich in dissolved iron. This low-energy environment constrained what life could do. Large, active bodies require oxygen. The Ediacaran world couldn't support them. Then, at the boundary between the Ediacaran and the Cambrian, the entire ecosystem vanished. The quilted organisms disappeared from the fossil record. Their extinction was total, and it cleared the stage. The Cambrian period begins 541 million years ago, and its start is marked not by a body fossil, but by a trace fossil, a record of behavior. The fossil is called Treptichnus pedum, and it's a burrow. Not a simple tunnel, but a complex, branching, three-dimensional network dug into the seafloor. To create this pattern, an animal needed a distinct front and back, coordinated muscle control, and a nervous system capable of executing a search program. Probe, turn, dig, move forward. This was an animal actively hunting for food buried in the sediment. It represents a fundamentally new mode of life. At the same time, geochemical evidence from rock cores reveals a spike in oxygen levels. The exact magnitude is debated. Some studies suggest a dramatic rise, others a more modest increase, concentrated in shallow coastal seas. But the trend is consistent. Oxygen concentrations crossed a critical threshold. Large active organisms organisms burn energy at high rates. Predation, sustained movement, and complex sensory processing all demand oxygen. The Ediacaran atmosphere couldn't fuel that kind of life. The early Cambrian atmosphere could. Calcium also increased. The concentration of dissolved calcium ions in seawater rose during this period, likely driven by increased volcanic activity at mid-ocean ridges, or the weathering of fresh rock exposed by melting glaciers. Calcium carbonate and calcium phosphate, the raw materials for shells and skeletons, became abundant and metabolically cheap for organisms to use. The environment didn't cause the Cambrian explosion by itself, but it removed the barriers. For the first time in Earth's history, the physical and chemical conditions permitted energy-intensive, armored, active life. Evolution had new options. All complex animals, insects, fish, birds, mammals, share a set of ancient genes called Hox genes. These are master control genes that regulate the layout of the body during embryonic development. They determine where a head forms, where limbs grow, and how segments are organized along the body axis. Hox genes don't build structures directly. They act like an architect's blueprint, telling other genes when and where to activate. A mutation in a Hox gene can shift an entire body part, placing a leg where an antenna should be, or duplicating a segment. The key insight from molecular biology is that Hox genes are old. The basic toolkit existed before the Cambrian, likely in the common ancestor of all animals. But at some point in the Precambrian, these genes duplicated. Gene duplication is a common evolutionary event. DNA copying errors sometimes produce an extra copy of a gene. Usually, the duplicate is neutral or harmful and disappears. Occasionally, it's repurposed. When Hox genes duplicated, evolution gained more instructions to work with. A larger genetic toolkit allowed for more complex body plans, additional segments, specialized appendages, distinct body regions. The explosion of animal diversity in the Cambrian may have been made possible by this expansion of the developmental programming language. This is the long fuse hypothesis. Molecular clocks, which estimate divergence times based on DNA mutation rates, suggest that the ancestors of major animal groups split from one another well before the Cambrian, possibly in the Ediacaran or earlier. The genetic capacity for complexity was already present. What the Cambrian represents then is not the origin of animal genes, but the ecological moment when those genes were finally expressed in large, hard-bodied, diversified forms. The fuse was long. The explosion was ecological. Predation changed everything. For most of life's history, organisms absorbed nutrients or filtered tiny particles from water. Feeding was passive. The Cambrian introduced a new strategy. One organism actively hunting, killing, and consuming another large organism. The evolutionary consequences were immediate and self-reinforcing. When the first predator appeared, any prey with even a slight defensive advantage, thicker skin, a hardened plate, survived more often. Natural selection favored defense. But once prey had armor, predators faced new pressure to overcome it. Selection favored stronger jaws, sharper claws, better senses. This is a coevolutionary arms race, and it escalates rapidly. Defense drives offense. Offense drives better defense. Each innovation by one side creates pressure for innovation by the other. The fossil record shows this sequence. The earliest Cambrian rocks contain what paleontologists call the small shelly fauna, tiny mineralized fragments scattered across the seabed. These are cap-like shells, spines, tubes, and plates, often disarticulated and difficult to assign to specific animals. They represent life experimenting with biomineralization before full-body armor appeared. Within a few million years, the experiment succeeded. Trilobites, arthropods with segmented, jointed exoskeletons, spread across the ocean floor. Shells appeared in multiple lineages. Mollusks, brachiopods, early echinoderms. Defensive spines became common, and then predators evolved to match. Anomalocaris, the abnormal shrimp, was the first apex predator. It grew up to a meter long, enormous for its time. It had large stalked compound eyes for tracking prey, two grasping appendages lined with spines to grab and manipulate victims, and a circular mouth made of overlapping plates that could crush shells. Fossils of trilobites bear bite marks consistent with anomalocaris attacks. The ecological feedback loop was now self-sustaining. Complexity became the survival strategy. Passivity was no longer viable. The innovations of the Cambrian didn't appear in isolation. They compounded. Biomineralization provided structure. Shells and exoskeletons weren't just armor, they were frameworks. Muscles could attach to rigid surfaces and generate leverage. A soft-bodied worm can contract and elongate. A trilobite, with its jointed legs anchored to a hard exoskeleton, could walk, dig, and manipulate objects with precision. Vision amplified the arms race. Trilobite eyes were made of calcite, the same mineral as limestone. Each eye was a compound structure with hundreds of individual lenses, each a tiny crystal of rock. These eyes could form images, detect motion, and judge distance. Before eyes, the sensory world was chemical and tactile. Gradients of smell, shifts in current, contact with surfaces. After eyes, the world became visual and spatial. A predator could see prey from a distance. Prey could see a predator approaching. This forced the evolution of speed, camouflage, and behavioral complexity. Efficient digestion powered everything. The evolution of the one-way gut, a digestive tube with a mouth at one end and an anus at the other, was a metabolic revolution. Ediacaran organisms had sac-like guts, or no guts at all. A one-way system allows continuous processing. Food enters, is broken down as it moves through the tube, and waste exits. An animal with this system can eat while digesting, extracting far more energy from food. High energy output enabled motility. Specialized limbs, jointed legs, swimming appendages, allowed sustained directed movement. Predators could chase, prey could flee or burrow, and burrowing changed the world. When animals began digging into the seafloor, they churned the sediment, a process called bioturbation. This released nutrients, oxygenated deeper layers, and destroyed the microbial mats that had dominated for billions of years. The old, still mat-covered world was physically torn apart. New ecological niches opened spaces within the sediment, new surfaces, new food sources. The substrate revolution was as profound as the biological one. Our knowledge of Cambrian life comes from a few extraordinary fossil sites where soft tissues were preserved. These are called Lagerstätten, German for storage places. Rapid burial in low oxygen mud prevented decay, capturing not just shells, but eyes, gills, guts, and muscle. The two most important sites are the Burgess Shale in Canada, roughly 508 million years old, and the Chengjiang biota in China, about 518 million years old. Chengjiang is older, proving that the full complexity of the Cambrian ecosystem was already established within 20 million years of the period's start. Trilobites dominate both sites. These arthropods were staggeringly diverse. Over 20,000 species are known from the Cambrian alone. They ranged from tiny scavengers to mid-sized predators. Their success demonstrates that the arthropod body plan, jointed limbs, segmented body, hard exoskeleton, was a winning design from the start. Anomalocaris is preserved in exquisite detail at Burgess Shale. For years, paleontologists found its parts separately and misidentified them. Its grasping arms were thought to be shrimp. Its circular mouth was classified as a jellyfish, and its body was believed to be a sea cucumber. Only later were the pieces connected into a single, terrifying predator. Opabinia is smaller, but no less strange. It has 5 eyes on stalks and a long, flexible proboscis ending in a small claw. When it was first presented at a scientific conference in 1972, the audience reportedly laughed. It seemed absurd, but it's real, and it represents one of evolution's experimental body plans, a design that didn't persist, but shows the range of possibilities being tested. Hallucigenia earned its name. It looked like a fever dream, a worm-like body covered in pairs of spines and tentacles. For decades, scientists had it upside down. They thought it walked on stilt-like spines with tentacles waving above. New fossils and scanning electron microscopy revealed the truth. The stilts were defensive spines on its back. The tentacles were pairs of clawed legs on its underside. It was a velvet worm ancestor, and the revision is a reminder that paleontology is a process of refinement. Pikaia is easy to overlook. It was small, leaf-shaped, and not a predator. It swam weakly in open water and was likely prey for larger animals. But it had a notochord, a flexible rod running down its back. The notochord is the defining feature of the phylum Chordata. Pikaia is one of the earliest known members of the lineage that would, over the next 500 million years, give rise to fish, amphibians, reptiles, birds, and mammals. It's a distant ancestor, and its survival in the Cambrian gauntlet is the reason vertebrates exist. This story isn't speculation. It rests on 4 independent lines of evidence. Exceptional fossils from Lagerstätten preserve details that would otherwise be lost. Soft tissues decay within days under normal conditions, leaving only bones and shells. But at sites like Burgess Shale and Chengjiang, rapid burial in fine-grained, oxygen-poor sediment halted decomposition. Eyes, antennae, gills, and even gut contents were mineralized and preserved. Without these sites, our view of the Cambrian would be skeletal, dominated by trilobites and shells, with no knowledge of Anomalocaris, Opabinia, or the full range of body plans. Trace fossils capture behavior. Treptichnus pedum, the burrow that marks the start of the Cambrian, tells us about decision-making, muscle coordination, and nervous system complexity without showing us the animal that made it. Scratch marks around Kimberella fossils indicate active grazing. Bite marks on trilobite shells match the jaws of Anomalocaris. These traces are the only direct evidence for the substrate revolution, the moment when burrowing animals began churning the seafloor. Geochemistry reconstructs ancient environments. Scientists drill rock cores from seabeds that were deposited 540 million years ago and analyze the ratios of different isotopes, variants of elements like carbon, sulfur, molybdenum, and iron. These ratios are fingerprints of ancient ocean chemistry. They reveal oxygen levels, nutrient availability, and the presence of anoxic conditions. The data consistently show rising oxygen and calcium concentrations at the Ediacaran Cambrian boundary. Molecular clocks estimate divergence times. By comparing the DNA of living animals, such as a fly and a human, and counting mutations in shared genes, scientists can estimate when their lineages split. The mutation rate isn't perfectly constant, but it's consistent enough to provide rough dates. Molecular clock studies place the genetic origins of major animal phyla before the Cambrian, supporting the idea that the explosion was ecological, not genetic. Each line of evidence has limitations, but together they converge on the same conclusion. The Cambrian explosion was real, rapid, and transformative. The Cambrian explosion wasn't instantaneous, but in geological terms, 20 million years is a blink. If Earth's entire history were compressed into a single 24-hour day, the Cambrian explosion would take 7 minutes. The genetic toolkit, Hox genes, the molecular machinery for building complex bodies, existed before the Cambrian. The ancestors of arthropods, mollusks, and chordates had already diverged. The fuse was long, stretching back into the Precambrian, possibly tens of millions of years. But genes alone don't create animals. They need permission from the environment. The Ediacaran world, with its low oxygen and limited calcium, couldn't support large, active, armored life. The early Cambrian world could. When oxygen crossed the threshold, when calcium became abundant, and when the first predator appeared, the fuse ignited. The genetic capacity for complexity met environmental permission and ecological pressure. Defense drove offense. Offense drove vision. Vision drove speed. Speed drove better defenses. The feedback loop became self-reinforcing. Within 20 million years, nearly every major animal body plan had appeared. Trilobites scuttled across the seafloor. Anomalocaris hunted in open water. Pikaia carried the notochord that would become our spine. The oceans transformed from a quiet, mat-covered expanse into a dynamic, dangerous, competitive ecosystem. The toolkit invented in the Cambrian, shells, eyes, guts, limbs, nervous systems, has never been fundamentally replaced. Every animal since has been a variation on these themes. The Cambrian didn't just add complexity to life, it defined what an animal is. The arms race it started has never stopped. The Cambrian didn't invent life, it didn't even invent animals. What it invented was the rules, the fundamental logic that still governs how animals survive. Armor, vision, pursuit, evasion. The feedback loop that started 541 million years ago when the first first predator appeared has been running ever since. Every adaptation you see in the natural world today, camouflage, speed, venom, cooperative hunting, is a continuation of that original arms race. The ocean floor stopped being a passive mat and became a battlefield, and life has been competing on that battlefield for 500 million years. The explosion ended. The war never did.